Our blood is a remarkable fluid, a bustling metropolis of cells with diverse functions. Among these cellular citizens, red blood cells, or erythrocytes, are the tireless oxygen couriers. They navigate the intricate network of our circulatory system, delivering this life-giving molecule to every nook and cranny of our body. The continuous production of these red blood cells, a process known as erythropoiesis, is essential for our survival. Erythropoiesis is a tightly regulated process ensuring we have just the right number of red blood cells to meet our body's oxygen demands without clogging up the circulatory system. Too few red blood cells, a condition called anemia, can lead to fatigue, weakness, and shortness of breath. Conversely, an excess of red blood cells, known as polycythemia, can thicken the blood, increasing the risk of blood clots and other complications. This delicate balance of red blood cell production is orchestrated by a complex interplay of hormones, growth factors, dietary components, and environmental cues. Understanding the intricate mechanisms that govern erythropoiesis is crucial for diagnosing and treating various blood disorders and maintaining overall health. Imagine our body as a well-run factory, where the production line is constantly humming, churning out red blood cells to meet the body's oxygen needs. Like any efficient factory, our body has feedback mechanisms in place to ensure that the production of red blood cells is perfectly calibrated to demand. This is where the concept of feedback control comes into play. In the context of erythropoiesis, feedback control ensures that red blood cell production is directly proportional to the body's need for oxygen. When oxygen levels in the blood dip, perhaps due to high altitude, lung disease, or blood loss, a sensor system alerts the body to ramp up red blood cell production. Conversely, when oxygen levels are sufficient, the production line slows down, preventing an overabundance of red blood cells. This feedback system operates through two primary mechanisms: functional feedback and end product feedback. Functional feedback focuses on the oxygen carrying capacity of the blood. When oxygen levels drop, specialized cells in the kidneys detect this change and release a hormone called erythropoietin, which stimulates the bone marrow to increase red blood cell production. End product feedback, on the other hand, monitors the actual number of red blood cells in circulation. When red blood cell counts rise, it signals the body to reduce erythropoietin production, thereby slowing down red blood cell formation. In the intricate symphony of erythropoiesis, erythropoietin, or EPO, stands out as the maestro, the hormone that orchestrates the entire process. This glycoprotein hormone, primarily produced by the kidneys, holds the key to maintaining a healthy red blood cell count. The story of erythropoietin's discovery is a testament to scientific curiosity and ingenuity. In the early 20th century, scientists observed that people living at high altitudes had higher red blood cell counts than those residing at sea level. This observation led to the hypothesis that a humoral factor, a substance circulating in the blood, was responsible for stimulating red blood cell production in response to low oxygen levels. This elusive factor was finally isolated and identified as erythropoietin in the 1950s, opening up new avenues for treating anemia. Erythropoietin exerts its effects by binding to specific receptors on the surface of erythroid progenitor cells in the bone marrow. This binding triggers a cascade of intracellular signals that promote the survival, proliferation, and differentiation of these progenitor cells, ultimately leading to the production of mature red blood cells. The production of erythropoietin itself is tightly regulated by the body's oxygen levels. When oxygen levels drop, specialized cells in the kidneys sense this change and increase erythropoietin production. Conversely, when oxygen levels are adequate, erythropoietin production is suppressed. While erythropoietin is undoubtedly the star of the erythropoiesis show, it doesn't act alone. A cast of supporting hormones plays crucial roles in this intricate biological drama. Some hormones, like androgens, estrogens, thyroxine, and growth hormone, have a stimulatory effect on erythropoiesis, boosting red blood cell production. Androgens, primarily produced by the testes in males, directly stimulate erythropoiesis, explaining why men generally have higher red blood cell counts than women. Estrogens, on the other hand, have a more indirect effect, influencing iron metabolism and availability, which in turn impacts red blood cell production. Thyroid hormones, secreted by the thyroid gland, are essential for overall metabolism and play a permissive role in erythropoiesis. Growth hormone, produced by the pituitary gland, promotes cell growth and division, including the proliferation of erythroid progenitor cells in the bone marrow. However, not all hormonal interactions are harmonious. Some hormones, like cortisol, a stress hormone produced by the adrenal glands, can suppress erythropoiesis. Chronic stress, therefore, can contribute to anemia. Cortisol appears to interfere with iron metabolism and erythropoietin production, ultimately hindering red blood cell formation. Section 5. The building blocks of erythropoiesis. Dietary factors. Imagine erythropoiesis as a construction project, a cellular endeavor to build millions of red blood cells every minute. Just like any construction project, erythropoiesis requires the right raw materials, the essential building blocks delivered through our diet. Among these, iron, vitamin B12, folate, and protein stand out as crucial for healthy red blood cell production. Iron is the most well-known dietary component associated with red blood cells. It forms the central component of hemoglobin, the protein within red blood cells that binds to oxygen, allowing them to transport it throughout the body. Iron deficiency is a common cause of anemia, particularly iron deficiency anemia, characterized by small, pale red blood cells. Vitamin B12 and folate are essential for DNA synthesis, a process crucial for the proliferation and maturation of red blood cells. Deficiencies in either of these vitamins can disrupt DNA synthesis, leading to megaloblastic anemia, characterized by abnormally large red blood cells. Protein, specifically the amino acids that make up proteins, provides the structural framework for hemoglobin. Adequate protein intake is therefore crucial for normal red blood cell production. Other nutrients such as copper, vitamin A, and vitamin C also play supporting roles in erythropoiesis, influencing iron absorption and utilization. Section 6. Environmental and intrinsic influences on erythropoiesis. Erythropoiesis, while primarily orchestrated by hormones and fueled by dietary components, is also sensitive to environmental cues and intrinsic factors within our bodies. These factors can either bolster or hinder red blood cell production, highlighting the adaptability of this vital process. One of the most striking examples of environmental influence on erythropoiesis is altitude adaptation. As we ascend to higher altitudes, the air thins and oxygen levels drop. To compensate for this reduced oxygen availability, our bodies increase red blood cell production, enhancing our blood's oxygen-carrying capacity. This adaptation is driven by increased erythropoietin production in response to low blood oxygen levels. Chronic diseases such as kidney disease, cancer, and inflammatory conditions can negatively impact erythropoiesis. Kidney disease, for instance, can impair erythropoietin production, leading to anemia. Cancer and its treatments can suppress bone marrow function, affecting the production of all blood cells, including red blood cells. Intrinsic factors such as age and genetics also influence erythropoiesis. Red blood cell production generally declines with age, contributing to the higher prevalence of anemia in older adults. Additionally, genetic mutations can affect various aspects of erythropoiesis, leading to inherited blood disorders such as sickle cell anemia and thalassemia. Section 7. The impact of drugs and chemicals. While our bodies have evolved to regulate erythropoiesis with remarkable precision, external factors such as drugs and chemicals can disrupt this delicate balance, either stimulating or suppressing red blood cell production. Understanding these interactions is crucial for both therapeutic interventions and recognizing potential adverse effects. Certain medications like chemotherapy drugs used to treat cancer can suppress bone marrow function, leading to a decrease in red blood cell production. This side effect, known as chemotherapy-induced anemia, is a common complication of cancer treatment. Other drugs, such as those used to treat HIV/AIDS, can also interfere with erythropoiesis, leading to anemia. Conversely, synthetic erythropoietin produced through recombinant DNA technology has revolutionized the treatment of anemia, particularly in patients with chronic kidney disease. This synthetic erythropoietin stimulates red blood cell production, alleviating the fatigue and weakness associated with anemia. However, the use of synthetic erythropoietin is not without risks. It can thicken the blood, increasing the risk of blood clots, heart attack and stroke. Certain chemicals, such as lead and benzene, can also interfere with erythropoiesis. Lead poisoning, for instance, disrupts the formation of heme, a crucial component of hemoglobin, leading to anemia. Section 8. Conclusion: a delicate balance. Erythropoiesis, the intricate process of red blood cell production, is a testament to the remarkable orchestration of our biological systems. It's a delicate dance of hormones, growth factors, dietary components, and environmental cues, all working in concert to ensure our bodies have a steady supply of these oxygen-carrying cells. The master regulator, erythropoietin, primarily produced by the kidneys, takes center stage, stimulated by low oxygen levels and driving the production of red blood cells in the bone marrow. However, erythropoietin doesn't act alone. A supporting cast of hormones, including androgens, thyroid hormones, and growth hormone, plays crucial roles, while others, like cortisol, can suppress erythropoiesis. Dietary factors, particularly iron, vitamin B12, folate, and protein, provide the essential building blocks for red blood cell production. Environmental factors like altitude and intrinsic factors such as age and genetics further influence this process. Understanding the intricate mechanisms that govern erythropoiesis is crucial for diagnosing and treating various blood disorders, managing the side effects of medications, and maintaining optimal health. By appreciating the delicate balance of factors that influence red blood cell production, we gain a deeper understanding of our body's remarkable ability to adapt and thrive.